Hello dear friends, welcome to the part 2 of multiple choice questions. This is the question paper of the entrance exam for admission to MSc in Applied Microbiology, which was previously conducted by Banaras Hindu University. Now it is conducted by NTA under the CUET PG exam. This video contains the question number 51 to question number 100, that means 50 important multiple choice questions. And these questions are very important for preparation of your exam, so watch this video till end. In the part 1, there were the first 50 questions, the link for which you can get in the description box below and in the I button here. The description box also contains the link for the playlist in which there are similar videos on multiple choice questions which will help you in the preparation of your exams and in your studies. Myself, Dr. Tripti Malika Ahuja. I am a tutor, guide and mentor for the subject microbiology, biology, life sciences and biological sciences. If you're new to this channel, then do subscribe it and also press the bell icon so that you can get the notifications of all the new videos. So before I start reading the questions, I suggest you to take out your paper and pen as I will read the question and then the answers, the options. One option is correct. You can mark the correct answer on your paper. Then I will tell you the correct answer with an explanation. If you are in doubt, you don't disagree or you want further explanation, then you can write in the comments. Question number 51. Which of the following is a requirement for restriction by a restriction endonuclease type 2? 1 ATP, 2 magnesium ions, 3 ATP and magnesium ion, 4 ATP, magnesium and SAM. The correct one is the option 3, ATP and magnesium. ATP hydrolysis is required for translocating the DNA past the enzyme, and magnesium is required for the cleavage of DNA. Question number 52. DNA fragments obtained by direct chemical synthesis can be joined to vector suitably by 1 homopolymer tailing, 2 blunt and ligation, 3 linker molecules, 4 ligation of cohesive termini. The option 4, ligation of cohesive termini, is correct. The DNA ligase joins the annealed cohesive ends produced by certain restriction enzyme. Question number 53. Required number of recombinants to achieve 95% probability of including any particular sequence in a random human genomic DNA library of 20 kb fragment size is. The size of genome is given to be 2.8 into 10 to power 6 kb. The different options they are written here. Now there is a formula in which the number of the recombinants is equal to natural logarithmic of 1 minus p. P is the probability and here the value of the p comes to be 0.95 divided by natural logarithmic of 1 minus 1 by small n. The small n, these are the number of the fragments which can be produced, so that is 2.8 into 10 to the power 6 divided by the average size of fragment, which is the 20. So this values come to be 1.4 into 10 to the power 5. And when we put this value in this question, then the values it come to be 4.2 into 10 to the power 5. Next question number 54. Which one of the following is not a limitation for cDNA cloning? 1. Larger messenger RNA are difficult to deal. 2. All RNAs do not possess 3' poly A tail. 3. cDNA libraries are rich in 3' terminus sequence. 4. Presence of poly A tails in messenger RNA. The limitation can be said at number 2. All RNAs do not possess 3' poly A tail. Actually, all eukaryotic RNAs, they have the 3' poly A tail except some transcriptome. So the correct answer is the option 2. Question 55. DNA chimera is a term corresponding to 1. Multiple copies of DNA, 2. RNA-DNA hybrids, 3. Gene manipulation, 4. circular DNA. The DNA chimeras, these are the RNA DNA oligonucleotides. These are dimers which are developed to correct the point or the frame shift mutations in the episomal or the chromosomal copies. So the correct answer is the option 2, which is the DNA RNA hybrids. Question 56. Stringency control during hybridization of nucleic acids on membrane is commonly controlled by 1. Temperature, 2. Time, 3. Concentration of probe, 4. Length of the probe. The correct one is the option 1, which is the temperature. The high temperature and low salt in the buffers, it is required for the proper hybridization of the nucleic acids. Question number 57. Organisms growing under high pressure have increased proportion in the cytoplasmic membrane of 1. Protein, 2. Saturated fatty acids, 3. Lipids, 4. Unsaturated fatty acids. The option, which is the 3. This is the correct option. The conformational changes in the lipids, they are responsible for the high tolerance in the high pressure. So the option 3, lipids, is correct. Question 58. OMPH proteins is synthesized in 1. Barophil, 2. Thermophiles, 3. Acetophiles, 4. Alkalophiles. Here the correct option is the 1. Barophiles, the organisms which can grow in the high pressure areas, and these are the deep sea bacteria, for example Photobacterium species. Question 59. Trophosome tissue of tube worm living near thermal vents harbor symbiotic. 1. Hydrogen oxidizing bacteria, 2. Sulfur oxidizing bacteria, 3. Nitrifying bacteria, 4. Iron oxidizing bacteria. The correct one is the 2, which is the sulfur oxidizing bacteria. So these are the gamma proteobacteria and they are the symbionts, for example the Candida and Doriflia. Question number 60. Which of the following plays role in the carbon cycle under anoxic condition? 1. Homoacetogen, 2. methanotropes, 3. chemolithotropes, 4. green algae. Here the option 2, methanotropes, is correct. The bacteria and archaebacteria, they can use methane as the carbon source and convert it into the biomass under the anaerobic condition. Question 61. Delta G for overall reaction of methanogenesis, 4H2 plus CO2 forms methane plus 2H2O is. The different values are given. Out of this, the correct value is the option 2, which is minus 130.7 kilojoule per reaction. The methanogenesis is a major metabolic pathway. It leads to production of natural gas, and for any spontaneous reaction, the value of the delta G is negative. So option 2 is correct. Question 62. Bacterial fermentation of glucose released through cellulose hydrolysis in rumen produces unsaturated fatty acids, option 2 carbon dioxide, 3 alcohol, 4 volatile fatty acids. Here the option 3, the alcohol, is correct. Question 63. Conversion of nitrite to N2O or NO, nitric oxide, under anoxic conditions is carried out by 1. Pseudomonas, 2. Nitrobacter, 3. Nitrosomonas, 4 is the rhizobium. Here the correct one is the Pseudomonas, which is a denitrifying bacteria. The one example is the Thiobacillus. Question 64. Which of the following herbicides has longest persistence in soil? Option 1, 2,4-D, 2. Delafin, 3. Simazin, 4. Atrazine. Here the option 3, Simazin, is correct. Simazin, it persists in the soil for 3 to 6 months. The protein which plays in the attachment of rhizobium to plant root is 1. Adhesin, 2. Ricardacin, 3. Rhizobin, 4. Lecithin. The option 2, ricardacin, is correct, and it is a calcium binding protein. The question number 66. Microorganism attached to opaque surface such as soil can be seen by 1. Light microscopy, 2. Phase contrast microscopy, 3. Novarsky effect, 4. Scanning electron microscopy. The correct one is the option, which is the option 4. Scanning electron microscopy. Here the surface details of soil as well as the bacteria, they can be seen. Question 67. Which of the following control measures is designated to break the connection between the source of the infection and susceptible individuals? 1. Quarantine and isolation of cases and or carrier. 2. Destruction of an animal reservoir of infection. 3. Treatment of sewage to reduce water contamination. 4. Destruction of vectors by spraying with insecticide. Here the option 3. The treatment of sewage to reduce water contamination is the correct one. Question 68. In relation to bacterium's optimal growth requirements, which group would you expect to be most likely involved in decomposition of compost piles? 1 acidophile, 2 psychrophile, 3 mesophile, and 4 is the thermophile. The option 3, mesophile, is correct. The mesophiles, they initiate the compost formation in the compost piles, and they are the most efficient in the compost preparation. Then these are taken over by thermophiles. Question number 69. Dash is the process in which microorganisms are used as a food source, resulting in nitrogen and phosphorus mineralization. Option 1 is nitrogen fixation, 2 is ammonification, 3 is microbiology, 4 is eutrophication. Here the option 3, which is the microbiology, is correct. It is the feeding of the microbes by the animals, and hence there is the mineralization of the nitrogenous and the phosphorus compounds. Question number 70. A microbial community that develops in puddles in low areas and retained on the soil surface is called a zooglia. 2 desert crust, 3 mycorrhiza, 4 is the microfilm. The correct one is the zooglia. Zooglia is a mass of bacteria which adhere in a jelly-like substance, and these are retained on the soil particles. These are particularly important in the wastewater treatment. Question number 71. The nitrogen fixation form of the rhizobium bacterium is called a 1. Bacteroid, 2. Symbiosome, 3. Infection thread, and 4 is the T. plasmid. The option which is correct here is the option 1, that is a bacteroid. The question number 72. One of the best solutions to get rid of non-biodegradable waste is 1. Burning, 2. Dumping, 3. Burying, 4. Recycling. So this is the recycling, option 4. I hope you remember the 3R principle of the waste treatment. Question number 73. In nutrient cycles in general, minerals tend to be dispersed through 1. Plant action, 2. Surface and subsurface runoff, 3. Evaporation, 4 is the assimilation. The option which is correct is the option 2, surface and subsurface runoff, the runoff of the water into the other bigger water bodies or to the groundwater. Question number 74. Dash fixes carbon in green plant as simple sugar. This is a very simple, very straightforward question. The options are 1. Denitrification, 2. Photosynthesis, 3. Carbonation, 4. Hydrolysis. I hope you have guessed the correct option, and the correct one is the 2. Photosynthesis. Question number 75. Microbial diversity is reflected as variation in 1. Adaptation to environment, 2. Cell size and morphology, 3. Metabolic strategies, 4. All of the above. The microorganisms, they differ in all of these: adaptation to environment as well as their size and morphology, that is variable, and the metabolic strategies that are also different. So the option 4, all of the above, is correct. Question 76. Banthing plants are 1. Too small to be seen with the naked eye, 2. Terrestrial plants living adjacent to lakes and waterways. 3. Also called cyanobacteria or blue-green algae. 4. Aquatic plants rooted in the river or the lake bottom. The option 4, aquatic plants rooted in the river or lake bottom, that is correct. These are macrophytic angiosperms which include the green, blue and brown seaweeds and the sea grasses. Question 77. The exponential phase in the growth curve in batch culture is as dx divided by dt is equal to mu x, where mu represents 1. Concentration of microbial biomass, 2. Specific growth rate per r, 3. Refractive index, 4. Natural logarithmic. The correct one is the 4. Specific growth rate per r. X is the mass or the number of the cells, the biomass, and t is the time period. Question 78. Substrate accelerated death rate. Death occurs when a cell is transferred into 1. Nutrient deficient medium, 2. Acidic medium, 3. Basic medium, 4. Nutrient rich medium. So the correct one is the nutrient deficient medium. In prokaryotes and eukaryotes, when the cells they are exposed to the lesser of the substrate, that can limit their growth rate. The question number 79. Zinc is an 1. Essential element, 2. Trace element, 3. Microelement, 4 is the macro element. The option 2, trace element, is correct. They are required in very lesser quantity. So this is a trace element, the option 2. Question number 80. 1 mole of glucose via oxidative phosphorylation liberates 140 ATP, 230 ATP, 3 25 ATP, 4 20 ATP. The option 2 30 ATP is correct. Question 81. The destruction of microorganisms is described as a 1. Zero order reaction, 2. First order reaction, 3. Second order reaction, 4. Third order reaction. The option 2, first order reaction, is the correct one. 82 question, a very simple easy question. The baker's yeast is Saccharomyces ludwigi, 2. Saccharomyces mycoides, 3. Saccharomyces cerevisiae, 4. Saccharomyces ellipsoides. The option 3, Saccharomyces cerevisiae, is the correct one. The question number 83. Active inoculum will give 1. Log phase, 2. Lag phase, 3. Stationary phase, 4 is the death phase. The active inoculum that will give rise to the log phase. So the correct option is the 1. Log phase. Question number 84. Increase in population number of cells in a culture is known as 1. Biomass growth, 2. Population growth, 3. Cell growth, 4 is the tissue growth. The option 2, population growth, is the correct one. Question number 85. The Waldorf type fermenter is made up of 1. Carbon steel and Cladlin stainless steel, 2. Silicon and stainless steel, 3. Pyrex glass, 4. mica and stainless steel. So here the correct one is the option 1. The carbon steel is clad in the stainless steel. Question number 56. Which can be used as an antifoaming agent? 1. Media, 2. Oils, 3. Nutrient, 4. Amino acid. The option 2, oils, is correct. The oils, they are used to minimize the operational consequences of the foam formation. The foam is formed when the high protein media they are used. Question number 87. Penicillin is derived from 1. Penicillium notatum, 2. Penicillium claviforme, 3. Penicillium tyrosinogenum, 4. Penicillium glaucum. The option 3, Penicillium tyrosinogenum, is the correct one. Question number 88. Microbial population maintained in which phase for long time using the continuous culture system? 1. Lag, 2 exponential, 3 death, 4 is the stationary. Out of this, the option 2 exponential is correct. In a continuous culture system, nutrient availability is unlimited. So the cells, they are maintained in the continuous log or the exponential phase. Question number 89. Cell density in dash is controlled by increasing and decreasing flow of culture medium. 1 chemostat, 2 turbidostat, 3 continuous culture, 4 synchronous culture. The option 1, the chemostat, is correct. The fresh medium, it is continuously added, while the culture liquid, it contains the leftover nutrients and the toxic end products that is remaining there, that is removed there. So chemostat is the correct one. The question number 90. Animal cell media contains option 1 RBC, 2 WBC, 3 platelet, 4 serum. The option which is correct is the 4 serum. The serum, that is the source of the important nutrients and important elements, a number of the substances which are stimulatory for the growth of the cells. Question number 91. The common cause of foaming in formation in fermentation is due to 1 carbohydrate, 2 lipids, 3 proteins, 4 vitamins. The correct one is the 3 proteins. In the protein-rich medium, there is the foam formation. Question number 92. The process of fermentation is carried out in the absence of 1 vitamins, 2 sugars, 3 oxygen, 4 amino acid. The correct option is the 3 oxygen. Question number 93. Beer is most accurately defined as 1. A beverage containing alcohol and made from cereals and hops. 2. A carbonated alcoholic beverage flavored with hop compounds. 3. A beverage made by fermenting sugars derived from barley, adjuncts and hops. 4. A beverage made by fermenting sugars extracted from malted barley and flavored with hops. So here the correct option is the option 4. A beverage made by fermenting sugars. These sugars, they are extracted from the malted barley and these are flavored with the hops. Question number 94. Which one of the following is being measured to determine the bitterness level in the beer? 1 hop oil, 2 beta acids, 3 iso-alpha acids, 4 are the alpha acid. Here the option 3 iso-alpha acids is the correct one. 95. Medical microbiology is a branch of medicine. 2. A branch of microbiology. 3. A branch of medicine and microbiology. 4. None of the above. The option 3, a branch of medicine and microbiology, is the correct one. Question number 96. Which is not a gram-positive bacteria? 1 Staphylococcus, 2 Lactococcus, 3 Mycobacterium, 4 Escherichia. The option 4 Escherichia is correct because it is gram-negative. Question number 97. Which of the following is a rod-shaped bacterium? 1 Neisseria, 2 Enterococcus, 3 Clostridium, 4 none of the above. Here Clostridium, which is a gram-positive rod, that is the correct answer. Question number 98. What is pathogen? 1. A biological agent causes disease to the host. 2. A biological agent provides support to the host. 3. Both 1 and 2. 4. None of the above. The option 1, a biological agent causes disease to the host, is the correct one. Question 99. What is commensal? 1. A relationship between two organisms where one organism benefits but the other is unaffected. 2. A pathogen. 3. Relationship between two organisms where one organism benefits but the other one is harmed. 4. None of the above. The option 1, a relationship between two organisms where one organism gets the benefit but the other may or may not be affected, or it remains unaffected, is the correct one. Now the last question of this MCQ part 2 is the Question number 100, which is a commensal? 1 Staphylococcus, 2 Clostridium, 3 Neisseria, 4 E. coli in gut. So here I will say the correct answer is the 4 E. coli in gut. Actually, it could be Staphylococcus also, if it have written Staphylococcus epidermidis, because some species of Staphylococcus like epidermidis, they are found on the skin as the commensal, but here it is written E. coli in gut, so that is the correct one. So these were the 50 important questions which will help you in the preparation of your exam. So best wishes for your exam. Bye and take care. Stay tuned for the next video.